My name is Sanjay Sisodia. I'm a professor of neurology at the UCL Institute of Neurology. My special interest is in epilepsy, especially epilepsies that are difficult to treat and may not have a known cause at this time. This project is about trying to understand these difficult to treat epilepsies better in order to get better treatment options for the people who suffer from these conditions. And these conditions cause a lot of problems for people who have them, including, for example, an increased risk of premature mortality. Increasingly, genetics is revealing the causes of these difficult to treat epilepsies. But the problem that we face now is that the genetic sequencing we do produces a huge amount of data, and the challenge is to interpret that data. Interpret that data in order to understand which variants might be relevant for the patient's condition, for the patient's phenotype. The project that Andre and I are offering involves novel ways of looking at patient-related data in order to make the most sense of the genetic sequencing information that's becoming available. And this will be done in a large group of people with very well characterized epilepsy. And we hope that the work that you will be doing will help us understand the genetic sequencing data better and therefore eventually better understand each individual's epilepsy. I'd like to hand you over now to Andre, who will talk about the technical aspects of this project. Thank you.
Hello, my name is Andre Altman. I'm an academic in the Center of Medical Image Computing here at UCL. And my background is in computational biology and in recent years also in neuroimaging. And that's the link to Sanjay. So this project is about looking at how genetics changes how the brain looks and how the face shape looks. And what we were working here in this project is two different types of imaging data and genetics data. On one hand, we have imaging data that is brain imaging data, classical brain imaging data, and we also have face shape imaging data. And we have standard genetics data at the moment. And what we're trying to do, the engineering part, is essentially a machine learning problem. So we try to understand how what we see in the genome predicts or explains what we see in those images that we acquire. So, and in the first part, we will try to link genetic information to face shape information and also genetic information to brain imaging data. This will help us understand how these rare epilepsies, the genetics of those rare epilepsies, are expressed in the face and in the brain. A second aspect of the project is to also link the two imaging modalities, so try to predict when we have a face shape image, what changes occur in the brain. This would allow us to do non-invasive screening, for instance, of people, because we only have to take like a stereographic image of the faces, which is really non-invasive. The third part, the most challenging problem, is to link all these three types of data, these three pillars, to the genetic information and two imaging modalities. There exist a few methods out there that are capable of doing this, but this is research in its infancy. What we hope to find out is that with these machine learning models, to identify states that explain part of the image and part of the genetics data, and these states we hope correspond to specific subtypes in epilepsy. So in the end of the project, we would link those states that we identified in a data-driven way to clinical information and hope that we can make better predictions on clinical outcome and treatment options. UCL is uniquely positioned for doing this kind of project because we have excellent clinical collaborators that provide us with this unique data, and we have also very strong engineering to tackle those challenges in machine learning.